So in this video, we see 10th SSC Part 2 Meiosis process. So guys, you know that meiosis occur in germ cell. So germ cell means that simple that one form in a male and one form in a female. In male that form in a sperm, and in female that form eggs or ovary. So in this meiosis stage, this meiosis is completely throughout the two stages. Those two stages are meiosis 1 and meiosis 2. In meiosis 1, that recombination. You all know what is a recombination or crossing over. You don't know, don't take tension. We discuss now. That crossing over between homologous chromosome, and thereafter those homologous chromosome that not sister chromated. This also we see in this figure. So are divided into two groups, and thus two haploid cells are formed. This all process we see in this figure, that meiosis part 1. So look carefully and listen carefully that in this meiosis we see prophase 1 phase. This prophase 1 phase that divided into 5 steps. So in this 5 step we see one by one. So this this first step we call leptotin. So in this leptotin phase you can see that chromatin fiber like structure. This chromatin fiber structure you can see that red and blue color. So that is called chromatin fiber structure, that that starting the condensation of a chromatin fiber. And you also see that centrioles develop, and some spindle fiber structure you can also see. So this only happen prophase 1 in first phase, that we call leptotin. Then next, that prophase 1 second figure. In this second figure that called zygotine. So in this zygotine, what here happen? In this zygotine that chromosome develop, but you can see in this chromosome that single chromosome, means like one red and one blue. So this one red and one blue chromosome we call homologous chromosome. And you also see that centrioles that goes on opposite direction. You clearly see in first image that centrioles are like near, but here that centrioles goes on opposite direction. You can also see that chromatin fiber condense and form a homologous chromosome, and this homologous chromosome that we call synapses. And this synapses here form. And you also seen that in the figure number 3. So figure number 3 is a very important for meiosis part 1 phase, because all this meiosis 1 phase that happen in figure number 3. So that figure number 3, that name is a picotin. So what is a picotin? What will happen in this picotin? You all here that we see that crossing over between two homologous chromosomes. So here crossing over present. So you see this chromosome that crossing over. See here that red goes on blue and blue goes on red. So here we call that crossing over. So that's we call picotin phase, and here crossing over. So in this crossing over we call chiasma. So in this chiasma here present, and also that centrals are same side, not change their position. And same as it is that spindle fiber also show here. But step number 4, here some different form. First we see that step number 4 in prophase 1, that name is a diplotin. So in this diplotin phase you can clearly see what different in figure number 3 and figure number 4. So in this figure number 4 you can clearly see that central goes and opposite direction goes. And some difference see in also chromosome. So that is a diplotin phase in prophase number 4. And last phase is a fifth number phase. You can clearly see in this phase, first we see here name, that fifth phase name is a dikinosis. So in this dikinosis phase you can clearly see nuclear membrane are completely invisible, and spindle fiber are fully developed, we also see. So that difference we see in step number 5. So here prophase 1 done. Here prophase 1 done. In this prophase 1 to 5 step, there are third step are very important. Why very important? Because in this third step that homologous chromosome are crossing over. So crossing over is a very important, and this crossing over that we call non-sister chromatid. Why non-sister? Because their color are different, that's why we call here non-sister chromatid. Then next phase we see that metaphase 1. In this metaphase 1, these chromosomes are lie in equatorial plane, and spindle fiber we also develop, but here nuclear membrane are invisible, that we also see in this prophase first. But here only that chromosome, not chromosome, that homologous chromosome appear in a equatorial plane. So that's only happen here, that metaphase 1. In this anaphase 1, we can clearly see that centromere are split, and sister, not non-sister chromatid are separate and pull apart the opposite direction. We can easily see in this anaphase 1 figure. And last one is a telophase, when you can clearly see that these two cells are separate, and here nuclear membrane also form, and this spindle fiber are invisible, and central are also formed. So this is a telophase 1. So that's why we see that meiosis part 1. In next part we see meiosis part 2. So meiosis part 2 is a very easy step and same as a mitosis step. So we see in this video in the next lecture.